Hi everyone, this is Ratna. Welcome to Basics of Biotechnology. In this tutorial, I am going to talk about mutations. What are mutations? Mutations means any change in the DNA of an organism which are unique and heritable are termed as mutations. Mutations can happen anytime and in a cell. The changes can be visible to it may be too minor change. These mutations may be lethal or beneficial to the organisms. Mutations can rise from chemical modifications and UV radiations. There are spontaneous mutations and errors during the replication time. Moreover, these mutations can be reverted back to normal state and hence they are reversible. Mutations from somatic cells are never passed to next generation. If mutation happen in the somatic cell, it will never pass to any daughter cell, any next generation. It cause both negative it have both negative and positive effects on an organism. It makes see mutations are the solo reason for evolution and will remain the most important part of life. It does not occur to cause diseases, rather it helps to survive by creating new alleles. There are various types of mutations are there, such as point mutation. In this point mutation, there are various types are there, such as synonymous mutation, non-synonymous mutation, frameshift mutation, and missense mutation. Now we are going to talk about synonymous mutation. What is synonymous mutation? A mutation changes one codon into another. See, in this sequence, uracil is there, and it is changing into cytosine. But what is happening here is it does not change an amino acid. Codon is changing, but it codes for the same amino acid. Both sequences are coding for the same amino acid, so it has no effect on the protein. As I told you before, in synonymous mutation, the codon is changing, but it codes for the same amino acid, UGU and UGC. Both this in this the third codon is changing. U is replacing with C, but both are coding for the same amino acid cysteine. What is non-synonymous mutation or missense mutation? In this, changing an amino acid. See, missense mutation means codon for one amino acid is changed into codon for another amino acid. See here, uracil is changing into guanine. But here, not like synonymous, it is changing the amino acid also. So it may affect the function of the protein because it's changing the amino acid, so it will affect the protein. So it is called non-synonymous or missense mutation. Truncation or nonsense mutation. It means codon of for one amino acid is changing into a termination codon. See in the sequence UGU, U is replacing with A, adenine. So UGU codes for cysteine. Now it is changing into UGA, so it results in stop codon. So what happens is it affects the function of the protein and the protein will not be translated. So it is called truncation or nonsense mutation. Deletion mutation. Deletion mutation means deletion of one or more bases. In this sequence, see here, uracil is present, UGU, and it codes for cysteine. Here, UGG, U has been deleted from this sequence, so it codes for different amino acid. So then what will happen? It affects the function of the protein. If one or two bases are deleted, it changes the open reading frame of the protein. Usually results to premature stop codon. It causes premature termination of the protein. Amino acids downstream of the deletion are different. Insertion mutation. Insertion mutation means adding of one or more bases. In the previous slide, we have learned about deletion of mutation. In that, deletion has happened. But on the contrary, in this mutation, insertion is happening, like insertion of one or more bases. See the sequence UGUG. On the other hand, see here, UGUU. U has inserted, one base is inserted in this sequence. So if one or two bases are inserted, it changes the open reading frame. See, it goes for the different amino acid and it changes the open reading frame of the protein, and it sometimes causes the premature stop codon, I mean termination of the protein. Point mutation in splice site sequence. What is splice site sequence? Spliceosome mechanism is means removal of introns and joining of exons. Introns means non-coding sequence of the gene and exons means coding sequence of the gene. It removes the non-coding sequence of the gene and joins the coding sequence of the gene. If mutation occurs at splice site, what happens is it may result to loss of exons or the inclusion of introns, and an altered protein coding sequence may occur. Point mutation in regulatory sequence. Mutation regulatory sequence may cause so many problems. Do you know what exactly regulatory sequence is? It means enhancers and repressors. They are capable of increasing or decreasing the expression of specific genes within an organism. It helps in transcription. If at all the mutation occur in this regulatory sequence, what happens is it can disrupt the transcription factors binding sites and or create a new ones. Sometimes it can change the level of expression of the gene, and it can change the time of expression of the gene during development, and it can change the tissue in which gene is expressed. Repeat expansions caused by expansion in DNA repeats. This repeat sequence or a continuous run of repeated DNA sequence. The repeats change size due to problems in DNA replication, and it tend to be very polymorphic in population. Usually it is di or trinuclear but can be more. This is an example of repeat expansion. This is an example of a repeat expansion mutation. This is a normal phenotype and this is a diseased phenotype. See how the repeat expansion, the nucleotides are repeated continuously. See here. The expansion in DNA repeats has happened in this disease phenotype. There are various types of chromosome mutations out there, such as inversion, translocation, duplication, and deletion. Deletion mutation. Deletion mutation means the loss of DNA sequence. See this picture. This is a chromosome structure. Can you see this break in the chromosome in the Q arm? If this part is deleted, then it will change the size of the chromosome. Because some part of the chromosome is deleted, so it will change the size, and sometimes it will lose the centromere position, and the functional genes involved in this part will also lose. There are some improved technologies to detect these micro deletions, and it plays an important role in pathology. Inversion mutation means one region of chromosomes is flipped and reinserted. See this picture. There is a break at the Q arm of the chromosome, and it is inverted and reinserted. Here in pericentric also, there is a break at the centromere position. It is inverted and reinserted. In both the cases, we can see if sometimes physically it can disrupt a gene, or sometimes it may have no effect. No associated phenotype apart from the observation that females with the inversion have more children. Translocation mutation. There are two types of translocation mutations are there. One is balanced and the other one is unbalanced. In balanced mutation, see chromosome A and chromosome B. There is a translocation between these two chromosomes. Chromosome AQ arm is translocating into chromosome BQ arm, and chromosome BQ arm is translocating into chromosome AQ arm. This is a balanced translocation. On the other hand, unbalanced translocation means this is a normal chromosome of parent 1 and the chromosome of parent 2 with balanced translocation. See this unbalanced one. It is getting one balanced translocation parent 2 and one normal chromosome of parent 1. So this is called unbalanced translocation. In balanced translocation, it retains the full genetic complement. It can result in phenotype. If break occurs in an important gene, it shows the phenotypes. Translocation have very useful in finding diseases. If translocation is present in all cells, problem will raise in the production of offsprings. Unbalanced translocation in gametes. Robertsonian translocation. It means it occurs between two acrocentric chromosomes. See this chromosome A and chromosome B. The Q arm of chromosome A and Q arm of chromosome B are fused together, and the short arms are lost. It contains only ribosomal genes, highly repetitive, therefore essentially functionally balanced translocation. It incidence in 1 in 1000 births. Significance is seen when these chromosomes segregate in meiosis. It may have an effect on potential offspring.